Good morning, sir.
Good morning, all of you.
Good morning, sir.
Okay.
Hello.
Good morning.
Yes.
Thanks. Now, today we'll have to discuss about the mutagen and the pharmacogenetics. Mutagen and pharmacogenetics. So, mutagens, that means these are the chemical agents or physical agents or biological agents that can able to change either in the chromosome number, or you may say that is the DNA structure, or you may say that is the DNA sequence. So by changing the chromosome number, by changing the gene sequence, or by changing the nitrogenous base sequence, mind that either changing the chromosome number or in the gene number or in the sequence of the gene, or you may say there is the nitrogen base sequence. When the characters, that is the genotypic as well as the phenotypic character of a particular species is changed, then this change is referred as mutation. And this is a generally a sudden change. Okay, then it is referred as mutation. And the agents which are responsible for induction of such mutation referred as mutagens. Referred as mutagens. And these mutagens are mainly divided into three categories. One is known as physical mutagens like ultraviolet ray, beta particle, gamma rays. Okay, these are all physical mutagenic agents. What are the chemical mutagenic agents? The chemical mutagenic agents like different drugs, different medicine, different drugs are also mutagenic agents. Different metal, like you may say arsenic, cadmium, nickel, chromium. Okay, these are also mutagenic agents. Drugs I have already mentioned, alkylating drugs, alkylating agents or alkylating drugs like cyclophosphamide, hydroquinone, these are also mutagenic. Okay. Another one, there is a biological mutagenic agents. Biological mutagenic agents, that means the different toxins coming from different bacteria, different some mushrooms, as well as some viral agents. These are also acts as mutagenic agents. So these are biological mutagenic agents. So there are three types of biological mutagenic agents. Okay, and we mainly discuss about the genes, genetic variation, and the drug efficacy. That is the pharmacogenetics. Okay, in connection with that mutation. Now, these mutagenic agents are also divided into several categories. Sometimes mutagens are also referred as carcinogens. All mutagens are not carcinogens, mind that. But the mutagens that have the capability for the resultant of cancer, that are responsible for the induction of cancer, these are known as carcinogens. Okay. So mutagens that can able to change the proto-oncogene into oncogene, which is responsible for cancer induction, proto-oncogene to oncogene. Or you may say that is the mutagen or the chemical substance that can able to suppress the tumor suppressor gene, that is the p53 gene, tumor suppressor gene or p53 gene. Okay. Then these are referred as mutagen as well as carcinogen. So one type of mutagen known as carcinogen that can able to induce the cancer by random cell division or uncontrolled cell division. How? Because you know that in cell proto-oncogene is present, and when the character of this proto-oncogene is changed and it comes to the oncogene, the presence of mutagenic agent, then the random cell division will be going on, and this random cell division or uncontrolled cell division and able to resolve cancer. Or other way, the mutagenic agent can able to suppress the activity of the P53 gene, like the tumor suppressor gene, that can able to arrest the random cell division, uncontrolled cell division. So if the activity, the suppression activity of the P53 gene is decreased, or another tumor suppressor gene activity is decreased, then the cancer is also noted. So mutagen can able to induce the cancer by two ways. One is by the conversion of proto-oncogene to oncogene, and number two, that is by suppression of the tumor suppressor gene like P53 gene. Okay. Now, number two, another mutagens are known as class 2 gene. Class 2 gene. What is class 2 gene? Now, mutagen that can able to reduce or that has the capacity to break the chromosome, known as class 2 gene. So some mutagen has the ability, some mutagens have the ability to break the chromosome, like beta particles, gamma rays. They have the capacity to break the chromosome. And due to the breaking of this chromosome, the gene sequence, the gene number will be altered and that results the mutation. And this is referred as class 2 gene. So if the question is given, what is class 2 gene? Then answer would be that some mutagens that have the capacity to break the chromosome. And as a result, it may induce deletion of some genes, because due to the breaking of this chromosomal arm, some gene will be deleted. So by deletion, by inversion, that is by changing the sequence of the gene. Suppose the one arm breaks or detached and then it is joined in other way. Okay. And as a result, the sequence will be altered. That is known the inversion. So by deletion, by inversion, due to the breakage of the chromosome, okay, then such mutagens are known as class 2 gene. Am I clear? Class 2 gene. Now, next one is that annulodogen. Annulodogen. What is annulodogen? Now, some mutagen can able to reduce the annuloploidy. Where is it? May reduce the annuloploidy. So annulodogen, that means they cause or they induce the annuloploidy. What is annuloploidy? That means they have the capacity to change the number of the chromosome. Not break the chromosome, but the number of the chromosome will be changed. That is the annuloploidy. Annuloploidy, that means when any species, if the number of the chromosome is changed, then this is known as annuloploidy. Okay? And then these are known as annulodogen. So these are the mutagens that can able to change the number of chromosomes. Okay? Now, some are also known as, some mutagens are present in inactive form, which are known as pro-mutagen. So pro-mutagen, which are present in the different foodstuffs, which are present in the different processed foods, which are present in the different preserved food, which are present in the different fast food, etc. The pro-mutagen, when are consumed from our body, okay, consumed in our body, then by different metabolic reaction, these pro-mutagens are converted into mutagen. And then this mutagen can able to change their effect. So what is pro-mutagen? Pro-mutagen, that means the precursor of the mutagen. Precursor, which are inactive form of the mutagen. Okay? And when we consume this pro-mutagen through our diet, then by biotransformation, which is mainly noted in liver, by biotransformation, these pro-mutagens are converted into mutagen. And due to such conversion, the mutagens can able to induce that effect. Okay. Now, next point that is very much important in connection to that mutation is known as silent mutation. What is silent mutation? Silent mutation, that means some mutagenic agents, okay, some mutagenic agents can able to change the nitrogen base sequence. But due to such change in the sequence of the nitrogenous base, okay, there is no change in the amino acid sequence. There is no change in the amino acid sequence. And as a result, there is no change in the correct phenotypic or genotypic character. Then these are known as a silent mutation. So mutagenic agent that can able to change the nitrogen base sequence in the DNA, but their effects are not expressed. Then these are known as the silent mutation. As for example, in case of the protein, due to the silent mutation, some amino acid are changed. But these amino acids are not the part of the active site of that enzyme. You know that enzyme has its active sites, either 2, 3, 4, up to 7. One enzyme may 2 to 7 active sites, which are responsible to bind the substrate and reduce the turnover from the substrate to product. So if the changed amino acid is not located in the active site, then the substrate binding affinity and its enzyme activity is not changed. But amino acid sequence has been changed due to change of the corrosion of the DNA. Okay, so this type of mutation in the genetical material is referred as silent mutation. So some mutagenic agent also result silent mutation, which are not expressed either in phenotypic manner or in genotypic manner. Okay, so this is all about the mutagens. Now we like to study, that is, the different drugs, how can able to induce the mutation. And next one, that is the pharmacogenetics, that means the activity of the drug. Efficacy of the drug, why vary from one individual to other individual, which is one of the personalized drug. Personalized drug. Okay. It is also differ from one ethnic group to other ethnic group. As for hydroxychloroquine for COVID-19, it may be effective for Indian or Asians, but it may not be active for other countries. Okay, like USA or UK, it may or may not be active. And just as for Asians. So that is the drug efficacy on the basis of pharmacogenetics. So first of all, we like to say that how the different drugs or medicine can able to induce the mutation. Now, different drugs like alkylating agents, like cyclophosphomides, hydroquinone, etc., they have the capacity to induce ROS, that is the reactive oxygen species production or generation. So alkylating agents have the capacity to induce ROS. And these reactive oxygen species are generated from uncoupling of the oxidative phosphorylation or electron transport chain, okay, electron transport chain, as well as microsomal oxidation reduction cycle. So these reactive oxygen species, which are known as hydroxyl radical, hydrogen peroxide radical, hydroperoxyl radical, superoxide anion, singlet oxygen, these are all reactive oxygen species. I repeat it, hydroxyl radical, hydrogen peroxide radical, H2O2 radical, hydroperoxide radical, HO2 radical, superoxide anion, singlet oxygen. So these reactive oxygen species generation in the cell is high. When the individual is subject under treatment of chronic treatment of alkylating agents, like I have already mentioned that cyclophosphamide, hydroquinone, etc. Now these reactive oxygen species can able to reduce the chemical change of the different nitrogenous base present in the DNA. As for example, cytosine. You know that in DNA, adenine, guanine, thymine, cytosine. So cytosine hydrogenous base present in the DNA would be changed into hydroxyl cytosine in presence of this reactive oxygen species, like hydroxyl radical or hydrogen peroxide radical or hydroperoxide radical. Okay, so cytosine transform into hydroxyl cytosine due to presence of the high level of ROS in the cell. Am I clear? Now you know that at the time of DNA replication, at the time of cell division, DNA replication is going on. So due to the DNA replication, you know cytosine always bind with or pair with guanine. And there is a triple hydrogen bond, GC, G triple bond C. So cytosine can able to pair with guanine, okay, at the time of DNA replication. But when cytosine is converted into hydroxyl cytosine, mind that, cytosine is converted into hydroxyl cytosine. This hydroxyl cytosine is unable to bind with guanine in the replicated DNA or newly formed DNA chain. In newly formed DNA thread, hydroxyl cytosine never invite the guanine. It will bind with adenine. So here, guanine is replaced by adenine in the new DNA chain or DNA thread. In newly synthesized DNA thread, mind that guanine bases are replaced by adenine because the DNA thread where the hydroxyl cytosine is present is unable to bind with guanine. It will invite adenine there and form a hydroxyl cytosine base pair. So guanine is replaced by adenine in the newly synthesized DNA chain. Am I clear? That means one purine base is replaced by another purine base, which is known as transition. So due to such replacement of one purine by other purine, that means guanine by adenine, okay? The nitrogen base sequence in the DNA of the newly synthesized DNA would be altered. And the three nitrogenous base in the DNA that represent the codon in the mRNA, that represent the codon. Codon, you know it, the three nitrogen bases in the mRNA that represent a particular amino acid in the translation process known as codon. So codon is present in mRNA, which are the three nitrogenous base sequence that represent a particular amino acid. Similarly, in DNA, the complementary nitrogen base of the codon are present. And these three complementary nitrogenous bases which are present in DNA respect to codon known as codogens. So in DNA, codogens are present, and in mRNA, codons are present. Okay, and on the basis of the codogens, codon are selected. Codon are framed in the nitrogenous base. Nitrogen bases in the codons are framed in the mRNA. So the controlling authority is the nitrogenous base in the codogen. I repeat it, codogens. What are codogens? Codogens, there is a three nitrogenous bases. Okay, summation or the combination of three nitrogen bases in the DNA that represent a particular codon in mRNA. These three nitrogen bases in the DNA known as codogen. So codogen, codogen, codogen are present, and in each codogen, three nitrogenous bases are present. Am I clear? So if the nitrogen bases of the codogens are changed, that is the guanine is replaced by adenine. Am I clear? Guanine would be replaced by adenine. So as the nitrogen base or nitrogen bases in the codogens of the DNA are changed, then nitrogen bases of the codon in mRNA would be altered. And due to such change in the nitrogen base of the codon, the amino acid sequence in the protein chain would be altered. And due to such alteration of the amino acid sequence of the protein chain, primary structure of the protein would be altered. And due to such primary structure altered, you know the activity of the enzyme, activity of the protein is mainly depends on their primary structure. So if the primary structure is altered, then the protein function will be altered. And that is referred to the mutant protein. If this protein is enzyme, then this mutant enzyme may or may not be able to perform their normal catabolic activity. If this protein is a carrier protein present in the plasma membrane, now this mutant carrier protein is unable to transport the specific biomolecule from inside of the cell to the outside or from outside of the cell to the inside. Am I clear? If this protein is actually a gate protein, gate like the sodium gate protein or potassium gate protein or channel protein, which is responsible to transport, that is influx of the different electrolytes or efflux of the different electrolytes. Influx, that means the entry to the cell, and efflux, that means expel from the cell. Okay, so if these are the channel protein, if these are the, you may say, gate protein, then such mutant gate protein, mutant, that means their amino acid structure has been changed, and due to such alteration of the amino acid sequence in the primary structure of the protein, their function will be altered. And now this mutant protein become unable, am I clear, to transport the particular electrolyte, to carry the particular electrolyte, And as a result, their functions will be altered. And as a result, their functions will be altered. Am I clear? So this is the basis of the pharmacological agent which acts as mutagen. So if the question is coming that pharmacological agents acts as mutagenic agents, justify their mode of action. So he also mentioned that mutagenic agents, especially pharmacological agent or the medicine which are mutagenic agents, which have the mutagenic activity, like the alkylating agents, like cyclophosphamides, which is immunosuppressive drug, which is also anti-cancer drug. Like hydroquinone, this is also one pharmacological agent having a mutagenic activity. And they also induce this mutation by the formation of reactive oxygen species. And these reactive oxygen species are singlet oxygen, hydroxyl radical, hydroperoxyl radicals, etc. What is their mode of action? Now, these reactive oxygen species can able to change the chemical nature of the different nitrogenous base of the DNA. As for example, you may say the cytosine, which is a pyrimidine nitrogen base of the DNA, in presence of RhoG would be converted to hydroxyl cytosine. And in the DNA replication, we know that cytosine pair with adenine, cytosine pair with guanine in normal condition. But when cytosine is transformed to hydroxyl cytosine, then in the replicated DNA thread, the guanine will not come. It will bind with adenine. So the guanine is replaced by adenine, and thereby the nitrogen base or nitrogen base sequence in the corrosion of the DNA would be altered. And due to such alteration of the nitrogen base in the corrosion of the DNA, the nitrogen base in the codon would be altered, because the corrosion is a controlling authority for the nitrogen base sequence in the codon. So due to the alteration of the nitrogen base sequence in the codon, the amino acid sequence in the protein chain will be altered. And due to such change in the amino acid sequence of the protein, that protein is known as the mutant protein. And that mutant protein, if it is enzyme, is unable to control such metabolic reaction. If this protein is a structural protein, am I clear? Structural protein, like it is a carrier protein present in the membrane, or it is a channel protein present in the membrane, or it is a gate protein present in the membrane, then it is unable to execute their specific function for transportation of the particular biomolecule to the cell or from the cell. Or if it is a channel protein or if it's a gate protein, then the mutant channel protein or mutant gate protein are unable to influx or efflux the concerned electrolytes, and thereby electrical potential of the cell would not be maintained. And due to such alteration of the resting potential, the cellular functions would be altered. The pH of the functions, pH of the cytoplasm would be altered, and due to such alteration, the cellular activity would be changed, and that is referred as mutation. Okay, so this is the mode of action of different pharmacological agents for the induction of mutation. Okay, now the next part, that is the pharmacogenetics, or what is the basis of personalized drug activity. Personalized drug activity. Find that. Personalized drug activity. That means one drug is very much effective for a particular group of individuals, but this is not effective in the other group of individuals. Why? That's the pharmacogenetics. That means the genetic setup of the individual will establish the efficacy, the activity of the particular drug, of the particular individual. And that is known as the personalized drug activity, personalized drug efficacy, personalized drug efficacy. And in society, it has been indicated that some drugs are very much effective in particular ethnic group, but this is not so effective in other ethnic groups. And that is the basis of the pharmacogenetics. This is the basis of the pharmacogenetics. Now, you know that the different drugs, like beta blocker drugs, like antihypertensive drugs, like antidepressive drugs, which are taken through oral route, are not present in the active form. These drugs undergo biotransformation in the mainly in liver, by an enzyme. There is a mixed functional oxidase system, MFOS, mixed functional oxidase system, MFOS, M for mixed functional, F oxidase O system, MFOS. Am I clear? And this MFOS, there is a mixed functional oxidase system. Out of this enzyme system, the most one important enzyme is known as cytochrome PD6. Cytochrome PD6. Write down it. Cytochrome, that is the CYP. Cytochrome P2 digit capital D6. Okay. Now, the gene of this enzyme is present in 22 chromosomes, 22nd chromosome of human being, at the Q arm, at the location of 13.1 position. So what is the location of CYP2D6 gene? 22 Q 13.1, mind that. So this is the location of this gene that is responsible for synthesis of cytochrome P2D6 enzyme. Okay. Now this gene is also present mainly in the two sets, but there are so many variations, 100 to 1000 variations are also noted. 100 to 1000 variations are also noted. Okay. Now the effective dose of the drug, toxicity of the drug, all are depend on the activity of CYP2D6, especially the beta blocker in respect to beta blocker, hypertensive drug, antidepressive drug, etc., etc. Okay. If the activity of cytochrome P or CYP2D6 gene, if the expression level of the CYP2D6 gene is high, I repeat it, if the expression level of CYP2D6 gene is high, then This enzyme in liver, which is a member of mPhos, that is the mixed function oxidase system. The activity of this enzyme, which is a member of the mPhos, is also high. And as a result, it can able to transform the inactive ingredient of this pharmacological agent, active pharmacological agent or active principle, into inactive to active very quickly and in most amount, that the quantity would be high. So the rate of transformation of the inactive form to the active form, rate of transformation as well as the quantity per unit time would be high. And as a result, the efficacy of such drug, like beta blocker drug, like antidepressant drug or antihypertension drug, in those group of individuals, okay, is very effective. That means sometimes standard dose may result toxicity in such group because due to the high activity, high expression level of CYP2 D6. And in those individuals, in those group of the individuals, okay, subtherapeutic level of this drug, subtherapeutic level of this drug is very much effective. Subtherapeutic level of the drug is sufficient for execution of their pharmacological activity. But if we provide the normal dose, it may result toxicity. And if we apply overdose, it may result some fatality also. Some fatality also. As for example, you know that hydroxychloroquine, which is given in patient, you know, some patients are also results fatality and some are not. And that is due to the variation of this member of the mPhos enzyme system. The individual where such member, that the cytochrome P26 enzyme, which is a member of the mPhos, is very high. The gene switch on level, the gene expression level is very high. In such individual, the inactive form is converted to the active form very quickly and it may result in fatality if such patients are under the treatment of normal dose. Okay, so dose selection, efficacy selection, that idea has been changed in the domain of the pharmacology. It should be gene-based drug standardization, gene-based drug efficacy. And that treatment profile will come in future and not at present. That means when the gene sequencing study, gene activity study on the basis of the Human Genome Project is very easier and we can able to do very easily on the patient. Then on the basis of this gene sequencing study, gene activity study on the human genome, thus those of the specific drug would be selected on the basis of that particular group. Okay, same dose would not be at present, you know, that same dose is applicable for all the individuals who are suffering from that particular disease. But at that time, the dose selection would be changed. Those are individuals are suffering from the particular disease, and that would be based on their genetic setup, on the basis of their rate of switch on or switch off of the particular genes which are responsible for the conversion of the inactive form of the drug to the active form. Okay, as for example, one example I also given here, that is the cytochrome P2D6 gene, which is located in 22nd chromosome Q1 13.1, which is the member, most active member of the mPhos. There's a mixed function oxidase system present in the liver. And this enzyme is mainly responsible for conversion of inactive phytoingredient, inactive pharmacological active principle into the active form. So inactive to active conversion of the pharmacological agent, which is going on in liver by mPhos. There are so many enzymes, there are so many members, and out of these CYP2D6 is the most effective one for the conversion of not all the drugs, but some specific drugs like beta blockers, like I have already mentioned that that is an antidepressive drug and that antihypertensive drug. And the group of the individual who had such gene expression level is very high. Okay. the normal dose of the drug may result the toxicity. And in such group, subtherapeutic dose is sufficient to overcome that pathological state. Okay. So this is known as personalized drug activity. It has been indicated that 5 to 10% population, 5 to 10% population of Asian countries where this CYP2D6 remain in overexpression state. 5 to 10% of the Asians where CYP2D6 remain in overexpression state. Overexpression. And thereby, if you provide such beta blocker antidepressive drug in those individuals at normal dose, it may result some toxicity. It may result some toxicity. Okay? And 1 to 2% of the individual, 1 to 2% individuals, okay, that have the CYP2D6 gene in ultra rapid state, that means their expression is very high. And in that case, half of the dose or 1/4 of the dose is sufficient for the execution of the activity. Okay, and this is the basis of the personalized drug activity or personalized drug efficacy. So today we finished that what is mutation, what are mutagen, what is clustogen, what is amyloidogen, what is mutagen versus carcinogen. And next point that we also covered that what is the basis of the mutation induction by some pharmacological agents or drugs. And last point that I have finished, that is the basis of personalized drug activity, personalized drug efficacy, which is the future of the pharmacology, future basis of the drug therapy, future basis of the drug therapy. Okay, that means on the basis of the DNA fingerprinting of the individual, when that fingerprinting is very much, gene sequencing is very much easy, okay, and it become user friendly, then that personalized drug treatment therapy will be introduced. So this is the future development of the pharmacology for assessment of the dose selection, efficacy selection, and toxicity of the drug assessment. Okay, on the basis of the specific gene expression. So 1 minute more. Is there any question?
স্যার এখানে যে ওই যখন ওই সাইটোসিনটা হাইড্রোক্সিসাইটোসিন হয়ে যাচ্ছে তখন কি ওই বন্ডিংটা জি এর সাথে সি যেটা ওটাকে ট্রিপল বন্ড
at the next time of cell division, it's the next type of cell division when this, suppose this is the DNA thread. Am I clear? This is the DNA thread, but this is the hydroxyl cytosine, and on that basis another DNA synthesis would be going on, DNA thread would be going on, another DNA thread would be synthesized, and at the time of synthesis nitrogen bases are coming. Okay, if it is cytosine, it will bind with guanine, but if it is hydroxyl cytosine, then it bind with adenine. As a result, the newly synthesized DNA, the nitrogen bases are changed. Am I clear? And as a result, the corrosion, there is the 3 nitrogen base in this DNA, the corrosion will be changed. Due to change of the corrosion,